Good afternoon, friends. And from the numbers, it appears that we'll be mostly rehearsing rather than presenting. Good, I have got some young friends behind from the undergraduate process. I'll try to make it exciting, and I will be able to relate to your subjects also. Will not be hovering only on clinical aspects, some basic science aspects also. As you know, mass has got an inherent tendency to give an impression, giving a survival advantage. Because in the process of evolution, when we started with the larger masses, it is aware that perhaps if you have a larger mass, you will have a survival advantage. But if you see the evolution of the nature, it has followed a principle of reduction in the mass, improvement in the design, and functional enhancement. So mass wise it has become smaller and smaller, design wise it has become better, and functionally it has become more efficient. That is why in nature all the big animals, they have disappeared, because you know the basic physics principle behind all survival is the principle of diffusion. Diffusion is dependent on surface area. When the mass increases by volume into density, that means 3 times, the surface area increases 2 times. If you remember the basic physics, pi r square versus pi r cube. So when the surface area is relatively becoming smaller and smaller, the mass will be reduced, because otherwise diffusion will not sustain for the life. So that is how we human beings has evolved. In 2002 and 2003, we had a challenge. We had a baby born with transposition of gate arteries. Transposition means normally in the normal heart, RV is connected to pulmonary artery and aorta is connected to the LV. But these babies are born with reverse connection. The blood vessels are transposed. So aorta is connected to pulmonary artery and the RV is connected to the aorta. So oxygenated blood is circulating on the right side in parallel with the deoxygenated blood, with the oxygenated blood circulating on the LV side, LV going to pulmonary artery, lungs and coming back. So these two parallel circulations are happening. So it is incompatible with life. Usually these babies die. And if we consider our population with a birth rate of 17 per 1000 population, now almost every 30 to 40 minutes, one baby is being born in India with these conditions. That roughly amounts to almost 10,000 babies. Now these 10,000 babies, if they are not taken care of, most of them they are going to die. So the days matters. And here was the challenge. The world order was that these babies can be operated maximum up to 2 to 3 weeks of life. Because when the pulmonary vascular resistance drops, the LV gets detrained. What is they call detrained? But the basic idea was the baby was born with a particular amount of LV mass, and because the pressure has dropped, afterload is raised, so LV starts losing mass. That was the basic premise. They thought that if they are doing arterial switch operation after 2 to 3 weeks, the LV will not be able to sustain the systemic circulation when it is connected to the aorta. So the idea was on mass, the restriction was the age. So as a bailout situation, they thought either you give an inferior type of surgery, which is called the SEMI, at arterial level you do the conversion, but you don't reconnect the aorta to LV and the pulmonary artery to RV. They said, okay, we can do pulmonary artery banding, create an narrowing by putting a band, so there will be a pressure load. Once there is a pressure load, the LV mass will start increasing. So they said we will go for a rapid 2-stage, band the pulmonary artery for some time and then do serial echocardiography, see how the LV is responding. After almost a week, you can go for a switch operation and LV will handle. But in 2002 and 3, we had a child who was 2 years old, PGA interventricular septum. nowhere in the world at that time a 2-year-old child with TGA intact ventricular septum has undergone arterial switch operation with regress ventricle. Nowhere. And that is the time I got an opportunity to do the surgery. I give my credits to my teammates and also my best critics. So I insisted that I will do arterial switch operation. The entire establishment, professional community said, no, you cannot do arterial switch operation because the baby will not sustain the circulation. Either you go for rapid 2-stage or you do a saline procedure. You cannot do switch operation. Here the journey started. And I hope many of you perhaps might have heard the name of Dr. P. Venugopal. He was ex-director and he was my mentor also in the department. So every evening we had Professor Anita Saxena, Professor Juneja, Professor Kothari, finest pediatric cardiologist, and Professor Venugopal, one of the aggressive critics and challenger. Plus I have my own colleagues. So every day I used to come in the evening and convince them that I want to do arterial switch. Then I started developing an idea. What is that idea? Because I am going to challenge all the boundaries that have been set in the world order. Nobody had done arterial switch at that age. Everybody thinking was mass is the most important thing. The only way you can go to the rapid 2-stage. So there is a law. There are 6 laws in biology in the process of evolution. The first law says that if Mother Nature wants to get anything done, then it must be done, and there are many different ways it can be done. So there is no impossibility. So you have to think in a different way. So there I put the argument. Now the topic started with regression. The world community was thinking regression means there is a waning of the muscle mass. But literally and evolutionarily, regression is something else. It has got nothing to do with mass. So they had the clinical community confused regression with mass. Regression literally means reverting back to earlier stage of development. Or regressive evolution means losing the characters which you don't require. You must have heard the species, those who develop in the deep cave underneath the sea, many kilometers down these things, because it is completely dark. So they lose the eye because there is no need for an eye. So that is called the regressive evolution. So I thought that the answer must be there in our past. Now you see carefully what is happening. I will go back to this picture. Here both the hearts are lying side by side. Topographically they are parallelly arranged. Am I correct? Topographically they are parallelly arranged. But circulation is in series, normal heart. Because RA, RB, PA, lungs, LA, LB, aorta, then back to RA. But when you come to the TGA, here the circulations are happening in parallel. There are 2 parallel blood flow happening and they are not mixing with each other except at the ASD level and at the PDA level. So we have topographically parallel heart, normal, and circulation in series. But in TGA we have a topographically parallel heart, but circulation is happening in series. Now go back to this. This is a very familiar diagram. My young friends must have seen in Gaitan, fetal circulations. You just see carefully that the left heart is pumping 58% of the circulating blood volume. RV is pumping 42% of the circulating blood volume. Exactly in TGA, the QP is more than the QS. So LP is handling more volume, though less pressure overload. But RB is handling less volume, but with high pressure overload. So exactly LB is connected to aorta, then aorta down below it is connected to placenta. Placenta is lungs, technically. It has a very low resistance. So technically LV is now connected to a very low resistance circuits. Like in TGA, LV is connected to PA and to the lungs. RV is connected to the lungs. Lungs has collapsed. So they have got high resistance. So typically physiology in baby is also in parallel and shunting is happening at the ASD and the PDL level or PFO level. I will let you think for a moment. How does the baby handles this circulation once he is born and comes out of the uterus? Think about it. I will give you the answer at the end, because here lies the answer to our entire effort. When the umbilical cord is clamped, the placenta is gone. Okay, saturation goes up, and the PFO closes, PDA closes. Now what happens to the circulation? It is converted to the series. So technically, physiologically, every one of us sitting here have undergone that arterial switch operation technically, physiologically. That by none other than most of the times in the villages by Dhai. So nature was so sweet that even it has endowed an ordinary person to perform the major task of converting parallel series to a parallel circulation to a series circulation. Okay. Now, when I insisted that we can do, if a baby can handle the birth, the 2-year child can handle. If I convert the outer to LV and PA to RV, I will switch over. Then I have to demolish the idea of banding. This is one experiment in 79. They have shown that when you give different loads to muscle fibers, the muscle fibers increases more force of contraction. Now, this is 1 to 8 weeks experiments, the top one, young one. You see up to 3 to 4 weeks, the predominant force of contraction increase is because of the neural adaptation without increase in the muscle girth. And if you see in the counter arm, there is no load given. The force of contraction is tremendously increasing. That means whatever muscle mass we have, just by neural adaptation, it can increase higher force of contraction. So there is no need to undergo hypertrophy. Because I told you, hypertrophy per se is a principle against the evolution. Wherever anything has hypertrophy, they die early. I will show you the latter part. Then you go to the old experiment. Muscle fibers are old, or patients are relatively older. Now we know that we are born with muscles fiber. It is fixed. They last to your end of a life. They are aging. So as you take older and older children, so the second graph shows that increase in the force of contraction that you are going to get maximum is out of the neural adaptation. This is just an experiment in the skeletal muscle. Now we have to demonstrate in the cardiac muscle. See, look at this graph. It is in the diagram. When you fill the LV with different volumes and close the aorta and stimulate the muscles, LV can generate a pressure up to 275 without increasing the muscle mass. And what pressure we are operating? 80 to 120. So that means mechanical efficiency what we have, we are most of the times in normal life working at 1/3. So we have got 2/3 reserve already. And this graph clearly shows just by having neural stimulation, the cardiac output from 5 liter, it can go up to 24 to 25 liters without increasing the muscle mass. And you must be knowing this is the Starling's curve. As the sarcomere length increases, usually sarcomere is from 1.6 micron to 3.2 micron. So as the sarcomere length increases from 1.6 to 2.2, the force of contraction increases. But when you go beyond 2.2 to 3.2, the force of contraction decreases. But we have not been given an accurate explanation why. We have been told that there is an appropriate overlapping of the actin-myosin bridge. But nobody actually explained with the stretching of the sarcomere, with every little stretch, the amount of physical overlap of the actin and myosin are gradually decreasing. The passive overlapping of the bridge actually decreasing. But when it is decreasing from 1.6 to 2.2 micron, why then the force is increasing? The answer lies here. The efficiency of the muscle fibers change. Now, I think MBBS students might be knowing that other than actin and myosin, we have an important fiber protein which is called titin. Titin acts as a springboard. It senses the tension in the muscle fiber. So once it senses the muscle fiber tension in the muscle, so it reconfigurates the actin myosin overlapping. Once that is reconfigured, the ATP hydrolysis increases. The calcium sensitivity increases. So that is why keeping the length constant, suddenly the force increases. Now coming to hypertrophy. This is I am trying to validate the idea that I don't believe in the mass theory. I don't believe in the idea of banding. I don't believe in the age boundary. So at an ideal level, I am validating from the existing knowledge from the basic science. Now I hope you know that the center of a cell from the nearest capillary is exactly calibrated and fixed all throughout the body, which is around 150 micron, except in the heart. The center of the cell from the nearest capillary is only 8 micron. And the density of the capillary network in the myocardial cell is maximum. So myocardium gets per gram per minute 0.5 ml blood. That is 5 times the liver. That is 15 times the skeletal muscle. That means it has a very rich supply of blood. And it is actually decreasing the diffusion distance. Because I told you that when the mass increases, the volume increases, the surface area proportionately decreases. Now, when the surface area is decreasing, we have to decrease the diffusion distance. So that is what the nature has done in the myocardium. But when you pack the muscle fiber with parallelly laid sarcomere, the distance between the nearest capillary and the center of the cell when the activity is happening is increased. The blood is not being adequately supplied. So with blood, oxygen is going. If you remember electron transport system, at the end, electron is received by the oxygen. Correct. Now, if it is not being received properly and oxygen is inadequate, then they roam around as reactive oxygen species and they actually damage. That exactly happens in hypertrophy. Hypertrophy muscles are inefficient. They die early. They undergo fibrosis. So there are 3 stages. And another interesting thing, that when you give a load to the myocardium, the intercellular matrix increases more than actually myofibers, because they have a very long half-life, and for bridging one peptide bond you need 5 ATP. That is the most energy expensive affair which a cell can spare. That is why protein is only used for building purposes, growth purposes. It is never used in the body for hypertrophy purposes, because once you use the protein for hypertrophy purposes, the entire purpose is defeated. And sarcomeres are constantly being formed and are being broken. So meaning thereby myofibrils are there, but sarcomeres forming are changing. You understand? So the net protein is constant, but they are being reconfigured from time to time depending upon the load condition. And this is again from the basic text. When the cells are starving for energy and you give a band, when you do a banding, when usually these babies are blue, and on the top of that you put a band on the pulmonary artery and the pressure load increases. So intracellular sodium level goes up, intracellular calcium level goes up. Once the sodium level goes up, you know that with each sodium 2 molecules of water go. The cells start swelling. The calcium remains inside the cell, and calcium knocks up the oxidative phosphorylation. So energy demand is increased, but energy production is decreased. So cells go in a suicidal pathway. So by principle, banding per se is not a physiologically adaptable or recommendable procedure. This gives you a final answer. Every one of you must have seen in Olympics that a 100 meter sprint runners, he runs and then at the end he squats down, or he lies flat, or he slows down. Otherwise, if he runs, then he will fall down. But if you see a sprint marathon runner, he keeps running. He does not fall down. So one indicates the power. The other one is indicating the efficiency. So now imagine heart is model for power generation or for efficiency. So now if you see the evolution also, when the baby is born, his heart mass to body weight is 3.3%. But when he is at 18 years of age, it is only 0.45% of the body weight. From 3.3%, the ratio is coming down to 0.5%. So mass wise it is decreasing. Now second thing, in the NB, again there is a further reduction. By 1 year of age, a baby weight increases by 3 times. The myocardial mass increases by 2 times. But LV mass increases only up to 1.5 times. So LV mass is relatively with aging becoming smaller and smaller in comparison to body weight and in comparison to the total muscle mass. So here is the crux. Whether the heart has been designed for endurance or for power, or it has been designed in favor to favor the mass or for efficiency. But till now you have seen it is designed for efficiency and for endurance. It is not for power, because we have got enough power. Now with this idea, when my colleagues got convinced, okay, I will thank Professor Chauhan, my teammate in the anesthesia and my perfusion team. So we took up that 2-year-old child for arterial switch operation. And when the child was admitted for 3 months in the ward, the entire debate, discussion and arguments were happening. All these things, whatever I showed you. This is the exact journey we have gone through. Then we were permitted to do arterial switch operation and we got successful. Without ECMO, primary ASO, 2-year child for the first time in the world. Now we want to validate the idea in a clinical setting. So we took up 11 babies, those who went for rapid 2-stage. Of course, this is by a different surgeon of my colleague, and 15 kids primary ASO, age group from 3 weeks to 8 weeks. And out of the 11 babies, those who had rapid 2-stage, 3 babies died after the first stage of PA banding and BT shunt. They did not reach the arterial switch stage. So that means, and then those who reached the arterial switch stage, out of the 8 remaining, 3 died. So success rate was 45%. But out of the 15 babies that we operated for primary ASO, 2 died, 13 survived. So it is 86% success rate. And in this setting, we operated 2 babies on integrated ECMO. You know the Helsinki declaration that whenever you offer any clinical treatment to a patient, patient's interest and welfare is the primary thing and safety is the most important thing. And for that we had ECMO in 2000 for the first time in the country. Institute had ECMO for the first time in the country in 2000. But from 2000 to 2005, we never had any survivor. I will show you. We had the first successful ECMO survivor in India in 2005. So we incorporated ECMO as a safety measure because we are venturing into a boundary where the world has not gone and we cannot compromise the safety. In the next paper, we took from 6 weeks to 9 years now. So we operated 55 babies. And out of 55 babies, 7 we lost. So with a mortality of rate of 12.7%. And when they died, Then we wanted to analyze the 7 deaths, why we lost them. And we analyzed that 4 we lost simply because they had sepsis. So everybody has an idea that if a child is not having fever or not having leukocyte count raised or CRP level is low, that means the baby is out of the woods and he can go for surgery. But we know from basic science that once a cell becomes sick, it remains sick at least for 6 weeks. Because the first thing that is impaired is the sodium potassium ATP system. Cells remain swollen and mitochondria are damaged. So energy production will not be there. So that is why you must have seen the babies, they look very dull, dusky, even if they don't have the fever. So when we saw that we have lost 4 babies because of sepsis and 3 more babies we lost because there was a delay in instituting ECMO. Because initially we used to operate in the OT and shift the baby back to the ICU, and when we see that babies are misbehaving, remember we are in a clinical course for the first time, we are not able to read what is the things happening. Our understanding of clinical behavior post switch was limited, and in the process after 4 to 6 hours or 7 hours when they used to fibrillate, that is the time we used to call pathologists. They used to come, they used to assemble the ECMO, and in emergency situation we put. So in the process what happened, we lost the critical part. And you remember that with every shock hypoperfusion, each cell in the body they lose adenosine, and within 30 minutes of cardiac shock they lose almost 50% of the intracellular adenosine. And if intracellular adenosine is not there, you know ATP cannot be found, and it takes 1 hour to recover 2% of that. That means 24 hours. So if 30 minutes of cardiogenic shock means 24 hours, the myocardium will not take the load because it is energy starving, and that is what we suffered. Because by the time we reinstituted ECMO, reinstituted the circulation, we have lost the battle. In the process, we lost 3 kids because of the delay in initiating ECMO, and 1 ECMO also related to infection. So in this situation, we found that wherever we predicted that the baby is likely to require ECMO, we did surgery on the ECMO circuit in the OT and kept them connected and off, and then shifted to the ICU, so that in the emergency, if the resident feels that patient is deteriorating, he has to just switch on. So we avoided crisis, and by not duplicating the hardware, because in the OT we are operating with 1 cardiopulmonary circuit, then in the ICU then another circuit, so cost was compounding. So we reduce the cost, we reduce the crisis, we reduce the chances of infection. So out of 7 babies, those who were post of ICU, ECMO institution, only 4 survived. Out of the 15 babies, those who had integrated ECMO, so 12 survived. So that validated integrated ECMO. That integrated ECMO, and this integrated ECMO concept and the circuit and the design, credit goes to the institute. It is our team, for the first time in the world, this has been designed. And this is now being extended to other areas of cardiac surgery where the babies are very sick. Then editorials were written on this article. Then we again went back to the subcellular level key, what is happening. You see one interesting thing, we got 20 babies dead, age matched heart from PGI Chandigarh, not for cardiac reasons, for non-cardiac reasons. Then we took biopsy here from our OT of preserved ventricle and regressed ventricle. Another one very surprising fact you see, between the normal baby, those who do not have any heart defect, their myocardial fiber count versus babies born with TGA, there is a significant reduction. But among the TGA, those who have preserved ventricle and regressed ventricle, their myocardial fiber count is almost equal. So that posed a fundamental question, that if with the same fiber preserved ventricle can come up bypass, so regressed ventricle can also come up. But the greater threat is that when these babies they grow up, they are virtually born with 50% reduction in the fiber count because cells numbers are fixed. So what is going to be their future life? But when it validated our idea, but it posed its subsequent question. And the babies, those who had, and you know that myosin has got two isoforms, alpha and beta, and they are equiposed, equally distributed. That also we demonstrated from genetic expression that both in regressed ventricle and preserved ventricle, both the isoforms are equally poised and equally distributed. And myosin heavy chain, it determines the amount of force the myocardium can generate. Myosin has got light chain and heavy chain. Light chain determines the speed of contraction, it regulates the speed, and heavy chain regulates the force. Heavy chain has got two isoforms, alpha and beta, okay, and that is also equally distributed. Then the babies, those who had surgery, we subjected to them to echocardiography and stress and strain analysis in different axes. We found that their post-op behavior subsequently on stress and strain analysis, there is not much difference, both in preserved and reserved ventricle, at rest as well as in stress. After confirming that, we wanted to know whether the coronary flow reserve is maintained. As I told you earlier, once the blood passes to the capillary in the heart, it extracts the maximum oxygen in one go because of the reduced distance between the center of the cell and the capillary. That is why coronary sinus flood is the maximum deoxygenated. The only way you can increase the oxygen supply by increasing the blood flow. So for that you have to see the reserve at rest and with exercise how much is increasing. And in this case both the preserved ventricle and regressed ventricle, it is maintained. Then subsequently we put them into adenosine stress test and the lab, and there also we found there is no difference at the end of 6 months and at around 6 years, because worldwide they had an incident that these TGA babies, those who have undergone switch, they either die in the immediate post-op because of coronary issue and bleeding issue, or they die usually 3 months again because of the coronary issue, or when they go to school playing. When they play, suddenly they fibrillate and they die. Nobody knew why they were dying. So that means when the exercise demand was increasing, the coronary blood flow is not increasing. That is why they were fibrillating. I wanted to know whether our kids are having the same situation, because clinically we never had any child when they were active playing had any problem. But we subjected him to adenosine stress test at the age of around 6 years. So there was no difference. And then we publish the midterm follow-up. They are the 55 babies with a mortality of 12.7%. And we said that most of the deaths are happening because of infection or because of the delayed institution of the ECMO. Delayed institution of the ECMO, we took care by integrating the ECMO. And regarding sepsis, that is the time when we took the help of pediatrics department, Prof. Of course we have got Dr. Menon. We got them inducted into the program, because every time we go, our colleague in cardiology, they used to say CRP is normal, count is normal, there is no fever, you go for switch. So we put our foot down, we said we are not going to do the switch because we don't have now age barrier. Let them recover, let them take 3 weeks, 4 weeks, 8 weeks, and then we will do the surgery. And one critical baby that time happened. The baby was born at the age of 29 weeks. Baby was 1.2 kilogram. Now remember the earlier idea that within 2 to 3 weeks of birth you have to do the switch. That is for 30 weeks birth babies. But when I was insisted that I have to do the switch, I insisted no, I will not do it. Let the baby complete the gestation period. So that means see, virtually I am going to spend another 10 weeks, means 2.5 months. After the baby is born, so LB will be regressed. So that is when Professor Paul ex-HOD pediatric medicine. He came to our help. He said, no, I will take care of the baby. Let baby complete gestation period. And by the time baby completed gestation period, he becomes 2.8 or 3 kilogram. And at the age of real 42 weeks, that means after 12 weeks of birth. So we did arterial switch and without ECMO, the child survived. So again, that broke the myth of gestation time and the timing of surgery. And in this case, the next 54 cases, we reduced the mortality from 12.7% to 3.7%. So what we propounded in 2009, that by taking care of reversible causes of sepsis, we will significantly reduce the mortality. That is what we demonstrated. So at that time, we had 556 babies. 185 was the regress ventricle. Of that, 57 were operated in integrated ECMO. Out of the 57, only 31 required, that is around 54%. But another interesting thing is that when we the team gathered the clinical experience, we could predict affirmatively which baby is likely to go for ECMO and which baby is not likely to go for ECMO. The babies which we decided will not go for ECMO and we did not do it on integrated ECMO, we were correct in 100% of the times. And in babies where we predicted that the baby is likely to go for ECMO, only half, 54% they require, but rest of the people required. But otherwise, if it would have been left to the previous mode of treatment, all these babies would have gone for rapid to stage. So that again broke the myth that even without ECMO, they are coming up bypass, only for safety sake. So we insisted that ECMO has to be there. So 12.7% up to 2009, then 3.7%, 9 and 10, and then it came down to 2.6%. And then we compared the children less than 3 months, but more than 6 weeks up to 12 weeks, or more than 12 weeks to 9 years. So there is no difference in the mortality rate. This is the current status. This is the world's largest series. When we published the first paper, that was the largest series, and today is also the largest series. And we have done now 763, of which 485 TGA IVS, of which 229 less than 6 weeks and 256 more than 6 weeks. Now, after validating the idea based on the basic knowledge, we validated it again on the clinical setting. Now we have to go back again to the cellular level to revalidate it or test it to make it foolproof. Now the fundamental question comes, how can the heart, by becoming gradually lesser and lesser in mass, can handle pressure more and more? Now we talked about efficiency. That efficiency comes from two sources. One, by the design of the heart. Second, by the energy metabolism. These are the two parameters that determines the efficiency. And you know that LV and RV are separated by interventricular septum. LV is exposed to high pressure, RV is exposed to less pressure. So it is one wall which is sensing both high pressure and low pressure. So this is the first wall which will shift its position and subsequently its motion. So that is what happened. When the IBS was protruding into the RV, the LV was spherical. Then gradually the IBS came to the midline. LV becomes D-shaped. When the IBS protruded into the LV, it becomes ellipsoid, banana shape. That exactly happens in pulmonary stenosis or ASD after a long time when the septum starts moving with RV instead of LV. This is the septum is the most important and most sensitive pressure sensor in the, it has a different anatomical design. So, but the Simpson method that is used by echocardiography friends, they will tell me, the Simpson method is based on assuming the transverse axis, equatorial axis as spherical, because it's you cut it as a consider it as a sphere, and like a stacks of coin, you divide the LV into multiple slices, and then you calculate their volume, and then you calculate multiply density, then you calculate mass. But now the LV has become spherical, banana shape. So the question comes, can it underestimate the LV mass? Now further, you know that a child has got a lesser body mass in comparison to body surface area. Now when it index it to body surface area, the LV mass is further underestimated. So we now gave a different proposition regarding regression in line with the evolution, that each regression is a programmed self-survival strategy, because now LV is not required to pump blood into the aorta because it's connected to pulmonary artery. So there is a less pressure, there is less energy requirement, it is handling volume more. So the cells are adopting that functionality without high energy demand, so that the cells can survive and at the same time carry out their function. So was it a programmed cell survival strategy? Exactly, the fetus, when it was going up to last 3 days of the birth, I am giving a leading it, up to last 3 days before the birth, it is also in a programmed cellular survival strategy on the cardiomyocytes. So it is not an atrophy. Then we went back to basic subcellular analysis. So we went for light microscope and also electron microscope study. On the light microscope study, we demonstrated they didn't have much difference between the regress ventricle and preserved ventricle. Of course, there are focal interstitial fibrosis, perivascular fibrosis, but there are evidence both on the regress and preserved ventricle, the evidence of cardiomyocyte hypertrophy. Hypertrophy basically we mean increase in the size. Okay, and how that size increases is happening, now we are not going back into the components. And if you see in a cardiomyocytes, normally mitochondria occupies almost 40% of the intracellular volume. Endoplasmic reticulum almost occupy equal amount of space, and there is one large nucleus. Now other than that, there are enzymes, electrolytes. For that space only 20% intracellular volume is available. The predominant volume is mitochondria and endoplasmic reticulum. So the answer lies in those two components. What is happening? Here is the answer. In the preserved ventricle, there is Z band is preserved, but in the regress ventricle, there is disruption of Z band, but there is no disappearance of Z band. There is myofibrillar disarray, but myofibrils are not lost. You remember in the earlier slides I showed you, when there is a load, the titin fiber actually reconfigurates the actin and myosin fiber. So they are well organized. When the cells are on high demand to generate pressure, so myofibrils are well organized, but not necessarily they are lost and over a particular sort of time. Then on the preserved ventricle, there are few fat vacuoles and there are glycogen granules. This is important finding. The entire fetus from start, from the tube state, it depends mostly on glucose and lactate for metabolism. Now, just before the birth, within 3 days of birth, it is shifting to oxidative phosphorylation. So because oxidative phosphorylation, it can produce large amount of energy. But we know glycolysis is with the fastest energy producing system. It can produce fast, but not high quantity. Efficiency is poor because it ends with pyruvate and lactate. And heartbeat, beat to beat, when the heart starts beating, before that intercellular pH is 7.3. and at the end of beating, pH drops to 7.1. So beat to beat pH variation is happening 7.3 to 7.1. So that is the amount of hydrogen ion which is being generated. And unless these hydrogen ions are washed up, the cells will not go for the next phase of contraction. That is why we have got a rich density of capillaries. So they immediately wash it away. So both the glycolytic pathway and fatty acid metabolism, oxidative phosphorylations are happening on the preserved ventricle. But once we come to the regressed ventricle, there is no fat there is plenty of fat vacuoles, there is no glycogen. That means these cells are not able to utilize 3 fatty acids and they are depleted of glycogen. The only alternate source which is there is actually reduced. So that means they are only purely dependent on glucose. There is no intracellular glycogen. If you cut off the external supply of glucose, that myocardium, particularly LV myocardium, will fail. And look at the mitochondria. Mitochondria on the preserved side, they are large, uniform in shape, and their picture-wise, their crystals are very well formed. But on the regressed ventricle, they are shrunken, non-uniform in size, plus sarcoplasmic reticulum. Sarcoplasmic reticulum works as a transport cell. Because ATP is being generated at one place, it has to be transported to some place. Nutrients have to be transported. They happen through these microtubules. When the energy demand is high, cells are very active. So mitochondria and sarcoplasmic reticulum are well developed. But in regressed ventricle, they are not well organized. So meaning thereby, when the LV was connected to low pressure circuit, he has shifted to a metabolic pathway which was there in the early phase of the development of the fetus. That is what is called regression. It has got nothing to do with mass. Now, this is a very simple lecture. I have taken it from Harper. My young friends will appreciate it. 80% of the BMR is consumed by sodium potassium ATP system just to maintain transmembrane potential. And whatever is little available for functionality, it is taken over by for the transport as well as for the contraction, locomotion. Now, last which is available is only for protein synthesis, peptides, except the enzymes. Because I told you each peptide bond requires 5 ATP. It's a very highly energy demanding. When the cells are starving for energy, obviously they will not make a ration for protein synthesis. So whatever in glucocardiography picture we are finding that LV mass is increasing by calculating the LV volume, myocardial volume, it is because of the interstitial edema. So this is in a nutshell, this is just a cellular adaptation, and glycogen is very essential to even to maintain contractility. And mitochondria draws 10% of its energy from glycolysis, 90% from oxidative phosphorylation. Of that 60% comes from free fatty acids and 30% comes from the glucose. When it is using free fatty acids, it will end in acetyl coenzyme, and acetyl coenzyme negatively inhibits hexokinase and phosphocutakinase. So that indirectly inhibits glycolytic pathway. So when there is high energy demand, it goes for a fatty acid metabolism, but keeps the glycolytic pathway as reserve. And when the energy is coming from straight from the glycogen, you know that we save one more extra ATP because there is no glucose phosphorylation required. So in the glycolysis we get 2 ATP, but if you get glucose from glycogen, we get 3 ATP. So that pathway is lost in the regressed ventricle. Now here comes the role of integrated ECMO. Why integrated ECMO was the game changer? Now when the patient is likely to fail and going to a cardiogenic shock, so the only pathway which was available by evolution is a glycolytic pathway. Fatty acid metabolism is not happening, PCA cycle is not happening, So only pathway which is happening is the glycolysis, which was 10% energy. It was just bare enough to sustain. But for that to continue, the hydrogen ion has to be washed out. So you need perfusion. A cell can survive with extreme hypoxia, but cannot survive lack of perfusion. So that there comes the role of the ECMO. ECMO maintains the circulation. It is normo-oxemic. It is normal temperature. The cells recoup. The mitochondria and the endoplasmic reticulum, they build up. And if you see their half-life, they invariably take around 48 to 72 hours. And in most of the cases in our series, we found that 24 to 48 hours of ECMO support is enough to bail the child out of that critical state. And if they succumb because of the sepsis, not because of the pump failure. And here also, now you know, these are the things you have to ensure to get the best result. And one of the interesting things that we started in 2003 is the enteral nutrition. Again, this is first time. There are literature that babies, when they go for surgery, they used to feed through Ryle's tube before surgery, or they have started feeding after 48 hours of surgery. But nobody has started feeding within 4 to 6 hours of surgery. Because the belief was, because they are under anesthetic drugs, there will be some problem. But when you saw while putting a PD catheter that there is resistance is maintained on the table, then we started feeding those babies after 4 to 6 hours. Now what has the game change which has happened? Because I told you, all these babies are dependent on glucose. They don't have glycogen stored. And usually sick babies in shock, they have got very little glycogen storage. The only way to supplement is by giving glucose. Now either you can give glucose IV, or you can give glucose feed mother's milk through enteral. And as you know, enteral feed is 1000 times better giving glucose. Because if you give IV glucose, the glucose fluctuation happens very fast. But with a enteral feed, that fluctuation does not happen. So in the process, so this is the fast baby on the left side, fast ECMO survivor of India. He is now 18-year-old. When he was operated with regress ventricle, he was 69 days old. This baby, when he was operated, he was 9 years old. Today he is 23 years old. And considering the entire research work, the AIIMS awarded this entire project as the path-breaking research work in the field of cardiovascular diseases for the year 2015. This is another baby. So the question is that when we have crossed the hurdle, there is only one leg stalk of the elephant. No, he has already crossed the barbed wire, but about to cross over completely. One of my very famous colleague who used to work with us here in the institute in Chennai, in one of the conferences, he asked, what is the most difficult thing of doing this kind of arterial switch operation? My simple answer was that, sir, switching of your mind. So the arterial switch operation has been done by Thais in the labor room. Mother Nature has done it. We can do it, but because we are stuck to our convention, that is why we are little scared to go beyond. But the beauty is that the world is now catching up. Last year, one of the center, they completely abandoned the rapid two-stage. They have gone full primary AS operations. So you must be knowing Watson and Crick. He said that if you want to really understand the function, then you have to go back and look at the structure. Without understanding the structure, you cannot understand the function. And for this entire journey, I thank my teammate, and they are from pediatric cardiology, cardiac anesthesia, my colleagues, pathology, pediatric medicines, and very well wishers, and particularly my residents. All these surgeries, path breaking surgeries I have done with the help of my residents. It is my residents, those who got me do these surgeries. So I give a special thanks to them, and special thanks also to my aggressive critics, those who challenged that you cannot do it also. Because if they would not have challenged, then perhaps we could not have done it. As the saying goes, kite rises against the wind, not in favor. If you want to fly high, accept challenges and go up. Now, if you don't take challenges, then you are destined to downfall. Thank you. Now it's open. You can ask.
You said in between your institutions were right regarding the need for it.
In entire my presentation, I did not insist on mass. You have seen. I did not insist, except that posterior wall thickness I used to ask. The most important thing I used to ask the diagnosis, whether it is CGR or not. Second, the position and motion of the IVS. Position means fast position wise it will shift, but till it is banana shaped, most of the times IVS starts moving with LV. When the LV is contracting, the IVS contracts with the LV. But when the regression happens further, intermittently it contracts with LV and intermittently contracts with RV. But when it's completely regress, then it completely works with RV. So any child who is banana shaped, but IVS is mostly or always contracting with RV, so that is the child is a sure shot case that will require, most likely to require ECMO support. But I told you, even when I took that clinical judgment, only 54% of those babies they require ECMO. That means the energy pathway is yet a mystery to understand fully, because there is no other way, because I cannot do an enzyme mapping of the myocytes before surgery to determine the metabolic. That it can be done from the myocardial tissue. You can map the key enzymes of the oxidative phosphorylation. From that index you can predict whether it is metabolically prepared or not, but that is not clinically possible. I cannot do a pre-op biopsy, but that part, but without compromising the safety, as I told you, IVS position and motion. I don't go for mass, I don't go for posterior wall thickness. We have operated with 2.2 millimeter posterior wall thickness. I have seen posterior wall thickness with 4 mm. But I will give you one interesting fact. When we are born, our posterior wall thickness say roughly 3.5 to 4. And when we grow up, by 18 years of age, our posterior wall thickness increases up to 8, 9, 10 or 11. So it is 2.5 times. Body surface area is 0.3. It increases to 1.6, 1.7. How many times? 5, 6 times. Okay. Body weight from 3 kg, it went up to 60 kg. So how much times? 20 times. But the irony, the beauty is that when a baby is born, his cardiac output is twice that of an adult. 3 kg or 3.5 kg with 550 ml of cardiac output. So roughly 150 ml to 200 ml per kg body weight. But in adult, it is only 80 ml, somewhere around 80 to 90 ml per kg body weight. As the mass increases, the amount of blood flow per gram of tissue which is required actually goes down. So the idea of having a powerful pump, having more muscle to generate high force so that we will overcome, is actually a myth. So because that is the limitation of our professional community, because we have a guild. मैंने आपने जो किया मैंने उसको देखा, मैंने वही किया। हमने एक दूसरे का किया, पर हमने एक दूसरे का वैलिडेट भी किया, अच्छा है बुरा है। हमने एक्सटर्नल वैलिडेशन कभी किया ही नहीं। हमारे पास एक्सटर्नल वैलिडेशन के लिए इन हाउस सबसे बेस्ट जो है बेसिक साइंस है, हमने उनके पास कभी गए नहीं है। सो टेक्निकली स्पीकिंग, व्हाट आई मीन टू से, I feel we can improve our outcomes if we can get ourselves back integrated into basic science. And all this work what has happened, we did. It's a leap of faith. But then for validation, we need Professor Panda who was there. We had a very good resident in pathology. When I wanted to get my electron microscopic reported, very surprisingly, it was very difficult to get it reported properly. And that resident who was a PhD student, he did that work for us. He is now in USA. And when we wanted a genetic this thing, that is when the Professor Panda came into the picture. And we went further even to check the apoptosis rate, because cells has come, because they said there is a waning of muscle mass. Then question comes, is there any loss of cells? Loss of cells can happen in three pathways: necrosis, apoptosis, and phagocytosis. Necrosis, electron microscopically showed there is no necrosis. There is stromal fibrosis, but there is no necrosis. Then apoptosis rate we compared equal, and phagocytosis we could not found in the electron microscope. So that all the three mechanisms for cell loss was missing in our electron microscope. So whatever cell were there, it was there, but cells were attenuated, means they were thin, because mitochondria content was less. So that is why I posed you a question: what happens? How the mother nature prepares the baby to be born? That answer we got in a 1912 research work. 3 days before the birth of the baby, the signal goes to the cardiomyocytes. The cardiomyocytes volume starts expanding. The surface area increases. Why surface area increases? Because receptor will increase. So the baby's heart is now going to an external environment to sense and process many signals, so for transduction. Then mitochondria, which was 10% in the baby, intercellular, jumps to 40% of this intercellular. Rough endoplasmic reticulum, which was around 10 to 50%, again it goes up. So that means the mother nature is preparing each cardiomyocytes so that when the baby will be born and the dye will clamp the umbilical cord, so the cells are energetically prepared to take that load. That is what we demonstrate. Thank you, everybody. Young friends, you want to ask anything? MBBS. Not the team. Not team. Compliment the team. You want to ask anything? I hope I have not bored you. Thank you.
Sir, I have. Suppose if you are like like 5 days.
I have operated on 21-year-old TGIBS.
Is there any predictor by which your ASO is going to be successful? Any predictors, echo-predictor, or like clinically? Because other easy of this go for setting with care and or trust. If you are leading into such high-risk cases, is there any predictors out of your clinical experience?
Predictors, I told you. I have given a slide on insurance. Okay. If IVS position and motion, you know. If you have gone with integrated ECMO, if you have gone with the close monitoring, and you can predict in the OT, IC. Usually, invariably, all these baby, you come a bypass on the OT. Then again, that gets to myth. Had the mass been less, then why did you come up to start with on the OT table when the SPR is maximum? And but when they come back to the ICU after 6 hours, 7 hours, 8 hours, or 4 hours, then 3-4 things happen. The baby, they will come, they will warm up, they will pass good amount of urine, pressure is fine, blood gases are fine. Then gradually the tachycardia will happen. And whose tachycardia, the most sensitive predictor is the arterial tracing. Remember I told you, because of that calcium mechanism, myocardial contraction versus skeletal muscle, the difference is that it is a slow contraction and a sustained contraction, unlike skeletal muscle. So arterial tracing, when the myocardial cells are contracting in a sustained manner, the arterial tracing will be different and the area under the arterial tracing will be more. But when it is reducing, then there will be a sudden rise in blood pressure, systolic blood pressure, it will not be sustained and it will drop down. So when the arterial tracing becomes narrow, even if systolic blood pressure is fine, tachycardia is happening, the child will start having fever, means low cardiac output, urine output start dropping, and then child will start developing acid-base imbalance. So that is the time before the electromechanical dissociation happens. So that is the time you have to go on ECMO. But when you go on ECMO, that itself is a separate topic. How to conduct ECMO in this situation. Most of the times you have a tendency of going full flow. But the moment you go on full flow, the artificial circulation has got a disadvantage. Correct? So you have to go with that much of flow, which will keep the heart beating, but the perfusion is going on. But if you go on full flow, heart is flaccid, then the complication of cardiopulmonary bypass will take over. So that is a clinical judgment. That is where the expertise of the team come. I know that you are now also doing it. So in our department now, most of my colleagues, Deva, Pradeep, Sachin, even Milind Hote, so they are doing it. And it's good results now. And this is a single surgeon's result that we presented.
How do you prognosticate the pregnant?
So we have done many times with fetal diagnosis is done. So the question is that in our country the irony is that there is no fetal screening. Rural population, and you know the high birth rate zone, UP, MP, Bihar and all those things, and there is no backup to make an early diagnosis. Then there is a delayed referral, and many times they come with chest infection, these things, and they pop up. Okay, now there are many like Saurabh and Rama, now they are focused and dedicated for pediatric cardiology care. But for that to come at a national level, it will take time. But when the fetal diagnosis is done, we have done it here. So we get the mother registered in the obs and gynae department. So babies it is better to transport a baby in utero than transporting a baby after being born. It is much safer and easier. So we get them registered here. We have even ladies from Russia. Those who came, got registered. They delivered the baby here, they had the arterial switch and they went back. But for that, you have to have fetal screening.
Last question. If you are fetal, is there any role of early intervention in or CTVS?
Being a clinician, I will not categorize it into early or late. I will rather put it in this way: appropriate intervention and timely intervention. Now, you should not go early out of panic. You should not delay out of your reluctancy. So for that, you must understand what is actually happening. You have to understand the pathophysiology first before you decide the timing and the nature of intervention. So deciding the timing and nature of the intervention, that is where it comes that a group of like-minded people. For example, in OT, I don't know. Remember, in the operation theatre, surgeon has a very, very limited load. The baby is completely under the custody of the cardiac anesthesia. No, I am lucky when the major part of the baby, safety and life, that is why there is a saying, Jamraj is only scared of anesthetist. because he is the only guy who can prevent death. It is not the surgeon. Okay. So when you have a very good anesthetist and he is fully committed and careful, you don't have to worry. Similarly, when you have got cardiologist, very good cardiologist, then automatically this decision. So everybody has to be good in that team. You cannot say I am good or I am bad, you are good, it can be compensated. In clinical practice, that is no compensation, that is called compromise. If the result has to be good, everybody has to be good. If one is bad, result is compromised. So regarding timing and nature of intervention, it is always the team approach. Then you have to see the comfort also. And most important thing is that why you want to intervene early? Why you want to have a particular intervention? I am very sorry to say that. Is it for patient or is it for yourself? If it is to your person, it is only for next day in a newspaper or it is for the patient. That decision you have to take. No procedure is done because surgeon wants it. But then if surgeon wants it, then he has to explain in the team and then you go for it. Yes. As I told you, in TGA, LV is handling high volume in comparison to RV. Technically, LV is having eccentric hypertrophy, not the concentric hypertrophy. Correct? And when you do a BAS, the LA gets decompressed. So pressure load on this thing is decreased. So they say there is a change in the shape. I will not say that by doing a BAS, the mass is reduced. The change in the shape happens much faster. If it is a circular, it will be D7. If it is a D7, it will become banana shape. But that does not necessarily correlate with the reduction in the muscle mass. No, look. Baby's recovery is not purely dependent on surgeon or the anesthetist. Okay. Earlier, when we were residents, all the babies, they used to be on Ryle steam before surgery. After surgery, they will go home with Ryle steam. Small, shrunken, dehydrated, alkalotic and dark. Because no internal feeding was given. And then we wanted how can we get rid of the simple most important factor for rapid recovery of a baby is the establishment of social communication between the mother and the child. And the best way to establish that communication is the feeding. So we encourage the baby, you should go for. So one of the approaches that the people said if you do a BAS, the LB will regress, then you should do an early surgery. But suppose mother has a cesarean section, he is in the hospital, there is no eye contact, child has not fed, mother's milk is not available. I will say do a BAS, saturation is improved, discharge the baby, let the mother feed, because I don't have a time boundary. When the baby comes back after 1 month or 1.5 months, he has got eye contact. So that is how in the OT, in the ICU, we allowed the parents to come and engage and spend time with the babies. So that helps in recovery. That outer boundary is that fear one has to get rid of. Okay. Thank you. Thank you.